Hello and welcome back to Teach Me in 10. In this episode, we're talking to Dr. Parashar Dhapola, CEO of Nygen Analytics. Parashar will introduce SiteType, a multi-agent AI system that transforms single-cell annotation for drug discovery by combining hypothesis generation, evidence collection, and expert validation all in one intelligent platform. Hi Parashar, how are you doing?
Hi, Laura. All good here. How about you?
Yep, all good, thank you. Thanks so much for joining us today. So we're going to be talking about the challenges of single-cell annotation and how SiteType is tackling them. So single-cell data is now routine in early discovery and translational programs. I wonder if you could start today by talking about at what point does annotation typically become the bottleneck and why does it persist?
Right. So that's absolutely true, Laura. Single cell has become a routine technique in drug discovery. It has really moved out of academia and has become the tool of choice for pharma companies to really look at their data at this higher cellular resolution. But here's the bottleneck, right? Once you have done your analysis, you've got your clusters, your trajectories of cells, then an expert biologist needs to come in and needs to connect those results with real biology. And that's the key. So the cell annotation process overall is not just about slapping some labels on the clusters, but really linking those clusters, those trajectories to underlying mechanisms, to generating functional insights, and doing it in a way that's contextual to your specific study. So if teams are looking at, particular patient samples, if they're looking at particular disease context or tissue context, it has to be relevant to those. So that interpretation work is where we are seeing that the computational teams and the expert biologist, they deliberated a lot, they have a lot of back and forth, and essentially the process is inefficient that leads to the whole drug discovery timeline slip little by little.
So essentially, teams are investing weeks, not just in the analysis itself, but in defending the calls they've made. Now, tools like Singular, CellTypist, and more recently GPT CellType each promise to solve this. Where do these approaches break down in practice?
Right. So tools like you mentioned, Singular and CellTypist, these reference-based tools are really good when you have highly curated, very high quality annotation reference available to you. In most cases, you don't, because the reality is that the samples will drift, right? Patients have different genetics, they have prior treatments, different disease stages where the samples come from, and also the tissue sources could be different. So what these tools, the reference-based tools, are trying to do really is they're mapping old knowledge to your new samples, and that's the antithesis of what novel discovery means. So the tools like others you mentioned, like the LLM-based tools, they promise to solve this because LLMs are really sort of adaptable to your context, to your cohort, and can do a lot of contextual assessment of your samples. But the current implementations have missed that opportunity. They're brittle, they hallucinate a lot, and essentially give you no evidence trail to link things back to. So regardless of the approach you're taking, these pharma companies are taking today, teams are still doing a lot of it through manual curation. And that step has simply not just gone away yet.
So really, with this manual curation step still being necessary, the bottleneck has just moved rather than been eliminated. And when annotation quality is poor, the effects aren't always obvious right away. What actually goes wrong downstream and how far does that ripple?
Right. So single cell data today, if you look at how pharma and biotech is using them, it's that those results are feeding into target selection, they're feeding into safety assessment, biomarker strategy. So let's say your annotations are, shallow or inconsistent. Those problems will not surface immediately, but few weeks later, a few months later, when a validation study fails, or when a reviewer asks for how did you come up with this particular annotation, so it's put in question and spotlight, that's where things start breaking. So let me give you an example. Let's say if you've labeled something as a macrophage, when really it is an M2 polarized immunosuppressive state, that's a completely different biological story because it's a potentially different target. So if your annotation is not that context aware, your biomarkers will not replicate, let's say, when you have new sample cohort or something like that. So that's one major part. The second part is just the day-to-day cost. You have a lot of people working on this through manual ways, and then there's a lot of cross-functional meetings happening to debate these annotation. A lot of time gets absorbed into that, and that is a lot of attrition and delayed times to decision. So this is essentially why we are focusing on annotation and looking at it as one of the hardest problems in the workflow and creating this next generation tooling to solve that. And SciType is all about solving that problem.
Right. So it sounds like the cost is real, both the hidden downstream risk and the day-to-day drag on teams. So let's talk now about how you approached building something better. SiteType takes a multi-agent approach instead of sending everything to a single model. Can you walk us through what that actually looks like step by step?
Right. So the we build SciType's architecture much like how scientists do their to take their annotation process. So you don't just simply look at a cluster and make a guess, right? You form a hypothesis, you test different evidences, you rule out the alternatives, and you document your reasoning along the way. That's the process we have built into SciType's architecture. So most LLM tools simply skip all that process. A lot of people have put a lot of faith on these LLMs that they will do all of this implicitly. We make that whole process explicit. So we don't just send the data to a model and say, what is this? But we try to create a trace that will give you the alternatives, that will give you the reasoning behind every action. And this is what makes SciType more appropriate than just for a quick glance. It's just giving you something that is defendable to collaborators that you can put in a regulatory package. And we use this so-called multi-agent orchestration to force this big scientific rigor in the annotation process. And we can broadly define that if you really go in deeper into 3 steps. So the first step is the contextualization step, where we build priors from your study design, your patient cohorts, your treatment history. If you're comparing treatment sensitive or treatment resistant samples, the model will understand that context and build that into the annotation itself. The second part is hypothesis generation and rejection. So we consider multiple candidates. We test them based on marker evidences, systematically rule out each one of them as they don't stand up to those evidences. And that creates a decision trail that will tell you why was it not something and why is it something. And the third part that we take is the literature linking. This is where we look at the literature, build confidence through that, try to look at market databases, external references, validate everything. And that allows us to surface that's something that's truly novel and not out there. So these 3 steps combined, when we have benchmarked them using different LLMs, not just the top-notch ones, we found that even weakest of the LLMs that are in the market can perform really, really well SciType due to its amazing architecture and doing it really, putting the scientific rigor in the process. And the results have been really good. We are seeing in the preprint that we released in November that SciType can do 300% better annotation in terms of accuracy in compared to ground truth against tools like SingleR and CellTypist.
Wow, that's amazing. And that decision trail that you talked about, it sounds like it addresses a real sort of accountability gap, like not just getting an answer, but being able to show you're working too. So the output isn't just a cell type label. What does a team actually get back from site type, and how does that change what they can do next?
So in all our conversations with pharma companies and biotech and trying to understand what they were actually doing, the fact is that most teams don't need broad cell type labels. They know their cells pretty well. And what they want to do is really want to go deeper into subtypes. They want to go deeper into cell states because that's what actually makes that data more actionable and relevant. So, and a cell type label will not get you there. So functional resolution is what you're trying to put into your data and understand from your data. So you need to surface not just like which genes are positive or negative, but which functional states are active, which pathways are active behind those states. And those are the pathways that will ultimately become targetable in your from your study. And that's what makes an annotation process really actionable. So for translational discovery teams, this changes the questions you ask. You're not asking which cell types are enriched in this particular case versus control. You're asking which functional states and programs are active and are they druggable. So for biomarkers, you would stratify by functional state, not just by cell composition, which can be very noisy. For safety teams, SciType can flag an inflammatory signature way before it becomes a problematic for downstream issues. So this is how your annotation can really be actionable for multiple teams across pharma, and that's where we feel like SciType can really make the annotation process more live and actionable than just static labels.
Parashar, this all sounds amazing. So for teams where this is a real bottleneck right now, what's the fastest way to evaluate site type on their own data?
The fastest way is to reach out to us and we'll run site type on your data. It could be one of your internal data sets or an external data set that you really care about. You get the results back, you evaluate them, see how they hold up to your own ground truth, see if you actually find something novel that you're really looking for in that data. And that's the starting point for a larger conversation of how we embed site type into your workflows. So it really produces the results in a format, in a way that is really attuned to your scientific process. And what we are already seeing with teams that have implemented site types, that they're saving weeks of times and getting higher confidence in their annotation process and moving that through regulatory process much faster. So you're not just debating labels in your meetings anymore. You're actually moving forward with your clinical, preclinical programs much faster. So if your annotation is, if annotation is the bottleneck when you're using single cell technologies, and for most teams it is, then do reach out to us and we'd love to show you how site type looks on your own data.
Lovely. Parashar, thanks so much for breaking that down so clearly. Really appreciate you joining us today.
Thank you, Laura. Thanks a lot.
Take care. Thank you for joining us for another episode of Teach Me in 10, brought to you by Nygen Analytics. Nygen develops AI-powered platforms for single-cell omics analysis, serving biopharma and academic research teams. For more information about site type, including a link to try it out for yourself, check out the links in the description. And if you enjoyed this episode, be sure to like and subscribe to catch all of our latest releases.